function away from insulin and glucagon and towards hormones that are important for growth and metabolism, and then water and electrolyte, salt and chloride and potassium and calcium control. So,
Bob, you're muted right now.
Oh, sorry, I muted it.
Thank you. Can you hear me now?
Yep.
Okay. We're going to focus on thyroid hormone, some growth hormone, and then some hormones associated with water and electrolyte control. That's where we're going for the next hour or so. So let's talk about thyroid. So thyroid hormones are amines. They're iodinated tyrosine derivatives. You need dietary iodine to make thyroid hormone. The stimulus is TSH from the anterior pituitary, which received its stimulus from the hypothalamus by the release of TRH into the portal circulation. So TRH from the hypothalamus stimulates the thyrotropes of the anterior pituitary to release TSH, which tells the thyroid gland to release thyroid hormones. The thyroid gland is the source of thyroid hormone, specifically the follicle cells of the thyroid gland. The targets are everywhere. The receptors for thyroid hormones that are expressed everywhere are intracellular, cytoplasmic receptors. They're not GPCRs, not tyrosine kinases. These are the intracellular ones that become transcription factors. And the effects are pleiotropic. There are multiple effects: cell respiration, metabolism, making of the sodium potassium ATPase, which you know is important for making gradients, solute gradients in the cells. We've used, remember that from the beginning of this course. Production of ATP by cellular metabolism. Production of heat, somatic heat, we make heat, and somatic development. So, multiple targets, multiple effects, all of them important. Here is the structure of T3. Two rings that came from tyrosine and multiple iodides, either 3 for T3 or 4 for T4. The thyroid gland is here in the throat, wrapping around your trachea. The production of thyroid hormones is an interesting story that I want to tell. So, where to start? Well, your book starts with the import of iodide, but let's just start with the production. Well, okay, there are iodide sodium co-transporters that are expressed in the follicle cells of the thyroid. So let's get oriented. Bloodstream, red blood cell, capillary, extracellular space, follicle cell of the thyroid gland, and then a lumen called a colloid. So this is not cellular, this is a space inside the thyroid. Cells, extracellular space, cells, endothelial cells, blood. Iodide is co-transported from the blood, and so it diffuses and then is pumped by a sodium-dependent iodide co-transporter. So remember, remember the sodium-calcium exchanger used the sodium gradient to pump out calcium. That was from the beginning of this course. This is the same sort of story, except it's using the sodium gradient to pump in iodide. So the iodide that is in small concentrations in the blood is pumped and concentrated into the thyroid follicle cell. The follicle cells of the thyroid make a protein by ordinary protein synthesis, ribosomes, etc. They make a protein called thyroglobulin. That's what TG is, thyroglobulin. Thyroglobulin is a protein. That's all it is. And it's a protein that can be exocytosed into the follicle, lumen, the colloid, and endocytosed from the lumen into the cytosol of the follicle cell. So there's this cycle of exocytosis and endocytosis of thyroglobulin going on all the time. Thyroglobulin is a protein. Some of the amino acids of thyroglobulin are tyrosine. So there's the tyrosine side chain of thyroglobulin. And there are enzymes, iodotransferases, that put the iodine that was pumped into the cell and put it onto the tyrosine and make monoiodotyrosine or diiodotyrosine. So these cells are specialized. They make they synthesize iodotransferases that iodinate tyrosine. And then they also make other enzymes that cut the iodotyrosine ring off of one tyrosine, and put it on the other. So we now make this two-ring iodinated structure on thyroglobulin. Thyroglobulin, whether it is iodinated or not, is cycle going on all the time. When stimulated by TSH, these cells express a protein that cuts here, releasing these molecules, T3 and T4, which are amphipathic into the blood. So this is a release story. This is maybe one of the few release stories that doesn't depend on calcium channels, calcium influx, doesn't depend on vesicular release. It depends on cutting and diffusion. It depends on the synthesis of iodinated thyroglobulin and then that cutting and release. It's a different story from just about all the other. We're going to see it one more time when we get to, we're going to see something like this when we get to steroid hormones. Okay.
I just have a question. When you asked about this story on an exam or something, when you ask about the protein synthesis and some things in yourself, is because you learned it, do you want us to repeat and know it, or can we just say
That's a good question. So the question was, if I ask you about this story, are you going to ask us to include details about protein synthesis, like mRNA and tRNA, and the tRNA, the peptidyl transfer, the thing that moves the amino-acyl group from the tRNA onto the newborn protein? Is that your question? No. I will not ask you about cell-phys questions that relate to. I will not ask you cell-phys questions related to metabolism, so I'm not going to ask you about glycolysis or Krebs cycle or electron transport chain. I went through it. I taught it again. But you've been tested on that. I'm not going to test you on that. And actually, protein synthesis isn't even cell-phys, that's CGI. So I'm not going to test you on CGI. It's there, it's important. I'm trying to emphasize that it's still important, but I'm not going to test you on it. I could ask you to tell me this story, though, knowing that there is a protein called thyroglobulin. Did that answer your question, Nick? So let's do this flow chart. Let's do this together. Okay? What's the name of the hormone that is released by the hypothalamus into the portal circulation that stimulates the thyrotropes of the pituitary, of the anterior pituitary, that stimulates the thyroid? What's the name that goes in here? TRH. Thyrotropin releasing hormone. What's the name of the hormone that goes in here? TSH. Thyroid stimulating hormone. Knowing that this is thyrotropin releasing hormone, and this is thyroid stimulating hormone, what's another name for TSH? Same story as with ACTH and CRH. TSH is also called thyrotropin. Thyrotropin releasing hormone. The hormone that causes thyrotropin to be released is thyrotropin. You don't have to memorize the names, thyrotropin or corticotropin. I'm just pointing out that the naming does in fact make sense if you know all the names. Thyrotropin releasing hormone, TRH. Thyroid stimulating hormone, TSH. What are the hormones that are released by the thyroid? What do we call them? I mean, we could call them thyroid hormones, but more specific. T3 and T4. And they have names too, triiodothyronine and tetraiodothyronine, but T3 and T4 is good enough. And now, so what are some of the effects? Pleiotropic, that's a word that I use. Pleiotropic just means many. Pleiotropic effects. What are some of the effects of thyroid hormone? I think it was on a previous slide, so just shout it out. This is not a trick question.
Increased cellular respiration.
Okay, respiration. Increased cellular respiration. Okay, that's another one.
Somatic development.
Okay, increased somatic development. Cell proliferation. Certain cells. I heard somebody say something, but I couldn't hear it clear enough. Is that you, Lindsay?
Low negative feedback.
Okay, so, well, T3, we're not getting to, so T3 and T4, so you're getting ahead of us. T3 and T4 do negative feedback. So high T3 and T4 would ordinarily do high negative feedback. Low T3 and T4 would inhibit negative feedback. So that's getting ahead of the story. We're gonna use that in a minute. So we just got a hint to a problem that's coming up. But what else do so T3 and T4 do negative feedback? That is, okay, let's include negative feedback. That is important. It's not one of the effects that It's not one of the feed-forward effects. It's a feedback. What's another feed-forward effect? Increases synthesis of the sodium-potassium ATPase, so it necessary ATPase for making and maintaining ionic gradients, homeostasis of ionic concentrations, and cellular respiration. Increased cellular respiration, actually, one of the byproducts of cellular respiration is heat, production of heat. So I don't remember what was on my slide. The next slide, that will be on Moodle in a few minutes, has these pleiotropic effects. Somatic development, respiration, sodium potassium ATPase, heat, somatic development, especially the nervous system. The nervous system is especially sensitive during fetal development to the presence of thyroid hormones. Okay, let's talk about hypo and hyperthyroid. Hypothyroidism. So what's the definition of primary hypothyroidism? Where is the defect in primary hypothyroidism? The thyroid. Primary hypothyroidism leads to low production of heat, so people are intolerant of cold, fatigue, weight gain because they're, well, fat is less sensitive, slow metabolism without decreasing appetite leads to weight gain. Appetite is a complicated thing. This bradycardia means slow heart rate, but you have to understand AMP2 to understand that, so we're not going to go there. One of the symptoms of primary hypothyroidism is goiter, of sometimes grotesque swelling of the thyroid gland. Big thickening of the neck because the thyroid gland gets huge. Question. Why in primary hypothyroidism, let's just say caused by an iodide dietary deficiency. Let's just take the simplest case of primary hypothyroidism. Low blood iodide because the person is not eating enough of it. The diet is deficient in iodine. Why would that cause goiter? Let's go back to this slide. Why would low dietary iodide cause swelling and enlarging of the thyroid gland? Goiter.
If there's no iodide in the diet, would you not have T3 and T4 to do the negative feedback?
Exactly. Low dietary iodide. You can't make the iodinated tyrosine derivative because you don't have the iodide. You can't make T3 and T4. You can't get this negative feedback. Excess, positive feed forward. Excess stimulation of the follicle cells of the thyroid to make the T3 and T4 that they can't make. Proliferate this positive arrow is not just telling the cells to make it, it's telling, okay, if you can't make it, let's make some other cell. Make a new cell that can make it. Cellular proliferation. Well, none of the cells can make it. because there's no iodine. So dietary iodide causes a huge, well, small or large goiter by lack of negative feedback and excess stimulation of the thyroid. Now, there's other forms of hypothyroidism. Hashimoto's disease is an autoimmune destruction of the follicle cells. So you would get all the symptoms, but you would not get goiter in this case, because if you destroy the cells, then they're not going to proliferate, right? The cells are gone. So you could have all of the symptoms of hypothyroidism, and then you could try to narrow down the cause depending on whether there was goiter or not. And now goiter is going to come up again because there is such a thing as hyperthyroidism. Sometimes it's a tumor of the thyroid that is telling, a cancer of the thyroid, proliferation of thyroid follicle cells due to cancer, proliferation of those cells is going to cause a goiter. But in this case, the goiter is associated with hyperproduction of thyroid hormone, not hypo. So it's interesting dichotomy for goiter. Thyroid tumor, goiter, production of too much thyroid hormone, too much heat production. People can't stand in a normal room. They're constantly, fanning themselves to try to cool off. Weight loss, metabolism is kicked into high gear. So no matter how much they eat, they're metabolizing it off too fast. They're releasing it as heat. Fast heart rate, anxiety, and nervous effect. There's an interesting phenomenon that I don't understand why, but the eyes tend to bulge out in people with hyperthyroidism. Something about the musculature and the fat behind the eyes pushes the eyeballs out. Pretty distinctive, actually. And so you can get it from a tumor of the thyroid, artificial stimulation of the thyroid by infection, artificial stimulation of the thyroid by antibodies. So Graves' disease, interesting disease, is a autoimmune production of antibodies that stimulate the TSH receptor. Antibody stimulation of TSH, so if we have antibodies that stimulate the thyroid gland, they're mimicking TSH. It's not TSH, but they're antibodies that mimic TSH. Then you get excess stimulation of the thyroid and goiter and T3 and T4 hyperproduction. So I do not recommend that you memorize symptoms. In fact, I request that you don't memorize symptoms. But I could ask you, if I could give you a problem, some text or something like I did on one of the exams, and ask you to interpret the text and apply this type of knowledge to answering a question. Okay, let's turn our attention to growth hormone. Growth hormone is a protein. It's released by the somatotropes of the anterior pituitary, and its release by the somatotropes is stimulated by growth hormone releasing hormone, and it's inhibited by somatostatin from the hypothalamus released into the portal circulation at the median eminence. The target cells for growth hormone are many: liver, muscle, fat, bone. During childhood and before puberty, growth hormone stimulates the osteoblasts and the osteoclasts to build bone. I'm not going to test you on this. Bone is one of those organs that I don't talk about a lot in this course, neither anatomically nor physiologically. Bone is an interesting organ. There are cells that make bone. Bone is a mineral, it's a calcium mineral, but it's built by cells. So growth hormone leads to growth of muscle, growth of fat, growth of bone. It's bone length that determines how tall we are. The receptor for growth hormone is one of the receptor tyrosine kinases, the story that you already know, that I hope you will tell me on the problem set and remember for an exam. Multiple effects, especially somatic development. But there is an interesting phenomenon with growth hormone. Growth hormone by itself has minimal effects. In order for growth hormone to do all of the things it's known to do, muscle development, bone development, growth, cell proliferation, it needs to synergize with another hormone called IGF-1, which is produced by the liver. So growth hormone works on the liver to produce another hormone that is necessary for all of the multiple effects of growth hormone. IGF-1 is a necessary co-enzyme, a co-hormone. It just is. For most of the effects. Not for the effects on the liver, but for the effects, other effects, especially on bone. IGF stands for insulin-like growth factor. So this is a growth factor that looks like insulin. And as a growth factor that is like insulin, it binds to, you can imagine what kind of receptor it binds to. It's a growth factor that's insulin-like, so it probably binds to a receptor that is like the insulin receptor, so it's another receptor tyrosine kinase. Okay, here's an interesting slide that I'm not going to test you on. It turns out, remember, that the hypothalamus really controls the release of TRH into the portal circulation, which controls the release of TSH from the thyrotropes of the anterior pituitary. So I hope you have annotated your slide here to say thyrotropes here, because I do want you to know the cell types that make the different hormones. So thyrotropes are the cells of the anterior pituitary that have TRH receptors and release TSH, thyrotropes. The hypothalamus is, it's integrating information from the body and from the environment. And one of the things that the hypothalamus does is it detects it knows when you're awake and when you're sleeping. Yeah. So, and you can see that the release of growth hormone is stimulated by sleep. So it turns out that children who are chronically sleep deprived or sleep interrupted, children who live in an environment where sleep is not a stable thing that is accessible, reduction of sleep, reduction of growth hormone, they tend to be small. Sleep interruption during childhood and puberty can inhibit growth by growth hormone. Here's a slide that I gave you with blanks in it. So this is using this homeostatic let's think about the homeostasis of the production of growth hormone. Stimulated by GHRH, inhibited by somatostatins. Antagonistic control of GH production. What goes in the box? Which hormone is stimulated if there is more than the set point amount of cell proliferation? What goes in this box? Or, alternatively, if there is lower than the expected amount of cell proliferation, what hormone goes in this box associated with this line? In order to maintain growth homeostasis, to release to achieve a set point of proliferation and growth. What goes in this box?
Somatostatin?
Let's see if this works. If growth is low, low somatic cell proliferation, do we want more somatostatin? I don't think so. What goes in this box? GHRH. If the amount of growth is low, we want to stimulate growth with GHRH. More GHRH than less somatostatin. Here is where somatostatin goes. Is that clear, Vincent?
Yeah, I think I was looking at it like when there is low cell proliferation, then growth hormone would be inhibited. But this is what you want to increase.
So that's an important point. This is not what causes cell proliferation. This is the response to this. So this is the response-dependent variable to the set point independent variable. If there is lower than expected cell proliferation, we want more cell proliferation to get to the set point, GHRH. Does that make sense? Sarah, you seem puzzled.
I think I was on the similar train of thought as Vincent.
Okay.
I've been thinking about the heater in the AC, like when there's higher temperatures, that air conditioner. I don't know. I think I was just thinking the same way as Vincent.
Yeah, I can see this is, sort of cause and effect issues, so we're talking about the effect on the hormone concentrations that would then bring us to the set point. So there's a mistake on the next slide. Here's the answer set, but this is not growth hormone. This is GHRH in here. GHRH would be high in order to raise growth hormone in order to increase cell proliferation. So I'm going to fix this before I post it. So don't copy this. GHRH is what goes in this box. Okay, in the past I've had a sort of a problem that we've discussed in this room. But everything's super fast and I'm mindful of time. I'm gonna gloss over this. It's an interesting problem. But, Hannah?
I was going to ask if this was the type of question, or if these are the type of questions you would ask in exams or quizzes.
I wouldn't ask this type of question on an exam. This is too philosophical for an exam. There's no clear right answer. So I wouldn't ask you to philosophize on one of my exams. Philosophy is important, of course, but not in this course so much. The point here is that there are people who, if their child is sort of on the small side of the growth chart, go to a physician and say, I don't want my child to be small. I want my child to be tall. I want my child. I'm raising a basketball player or whatever. I want my child to get growth hormone. In order, they've heard about growth hormone. They've heard about things that stimulate growth. Growth hormone injections, please. It's like, okay, that's probably not a good idea. I mean, it's certainly not a good idea. What test would you want to run? Well, what if the defect was not in growth hormone? What if the child had plenty of growth hormone, but was defective in IGF-1? Or maybe both of those are fine, and it's thyroid hormone. Or maybe all of that is fine, and this is just, wait a second. You're 5'9", your wife is 5'6", and your child is genetically the product of someone who's 5'9" and 5'6". What's the problem, right? And, so there's all kinds of issues, but physicians, endocrinologists, are often confronted with parents who say, my child's growing too slow, fix it. So it's a philosophical question. No, I would not ask that on you. So let's skip this and get to a clicker. Okay, let's do this one clicker. It's just one. Yeah, this is a general question. This is not just true about growth hormone. This is true about pretty much all of the stories we're telling. It is more difficult to measure GHRH from blood than it is to measure GH. It's just a trick. It's harder to measure. Why? Those of you who brought your clicker, go ahead and show me your answers or answers. Well, no, let's not. Let's just go through. Is this a true statement? No, this is not a true statement. They're both true. Is this a true statement? Yes, this is a true statement. GHRH is released into the portal circulation, a tiny little circulation between the hypothalamus and the pituitary. Growth hormone is released into the systemic circulation. So GH concentration, because it's going everywhere, or it needs to go everywhere to have all of its effects, is higher, much higher, in the blood that could be sampled. We cannot sample. So if someone says, well, just go into the portal circulation and measure the concentration there, that's so easy to say and it's almost impossible to do. You can't sample portal blood. You sample systemic blood. GH receptors, so B is true, A is false. C, growth hormone receptors are expressed on a wide variety of cell types. Well, that's a true statement, but it's not a true answer. That's a true statement, but it's not the answer to the question. GHRH concentration in the portal circulation is high, but GHRH concentration in the systemic blood is too low to be measured. Yes, that's a repeat of what I was saying. So B and D are correct answers to this quickly. Let's turn our attention for a few minutes, without a break, we're going to power through, to talk about some hormones associated with our ability to achieve homeostasis of water, salt, and calcium. To really do this, we have to understand the kidney. Those of you who took AMP2 understand the kidney. But I'm not going to teach the kidney now, because the kidney is AMP2. So I'm going to have to black box this a little bit. By black box, I mean not give you the molecular mechanisms, or only try to get the people who took AMP2 to remember. Let's talk about vasopressin and aldosterone for the control of water and salt concentrations in our body. We achieve homeostasis in the amount of water that is in our body. How? By drinking. Thirst is a physiological response to dehydration. Sweating, well, that's mostly heat, but that's a way that we lose water. Urination is a way that we excrete water. So we ingest water, we are homeostatically control the amount of water, and then we urinate water, either, or we don't. We don't urinate in order to maintain water. So, antidiuretic hormone, or vasopressin, where is vasopressin released? posterior pituitary. Its stimulus is osmolarity in the blood. So if you are dehydrated, too low, water is too low, your concentration of solutes in your blood goes up, the volume of your blood goes down. Both of those are detected by the brain, integrated by the hypothalamus, triggering the hypothalamus to release vasopressin from the posterior pituitary. Vasopressin is a peptide hormone. The target is the kidney, as I said. And now the effects. Those of you who took AMP2, vasopressin, GS coupled receptor on the cortical medullary collecting tubule, GS, PKA, phosphorylation of aquaporin, da 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 da. Those of you who didn't take AMP2, come back. The receptor is a GPCR, and it leads to water reabsorption. It allows the kidney to not excrete water. Thirst, drinking water, is a complicated thing that is also maintained by the hypothalamus. The hypothalamus has centers that trigger water seeking and water drinking in animals as well as humans. Vasopressin, water homeostasis.
Bob,
Yeah.
so I know when I get particularly anxious that my hands get really sweaty. Does that anxiety induce a hormone response that would cause that excess sweatiness to occur, or
Yeah.
release of water?
That's not that's sort of neuroendocrine. That is epinephrine and norepinephrine triggering your sweat glands. In fact, sweating on the palms, it's one of the things that is detected in the lie detector test. So they and over in the psych department they have tools. It's called the skin galvanic response. If you sweat salt water onto your skin, the resistance of your skin, the electrical resistance, goes down. The conductivity of the surface of your skin goes up because there's salt water on your skin. And so the skin galvanic response can be detected by measuring the resistance or the conductance of the skin. If you are nervous, it goes up. If you're relaxed, it tends to go down. It's a very slow response. It's pretty noisy, but it's used in the lie detector test. The idea is if you're telling a lie, you're getting nervous, your heart rate goes up, your blood pressure goes up, and your skin galvanic response goes up. But it's more a response to your sympathetic nervous system than it is to anything more endocrine than that.
Okay, thanks, Bob.
Okay, getting back to this idea of salt and water homeostasis, vasopressin is critical for water homeostasis. Aldosterone is critical for salt homeostasis. Those of you who took AMP2 know that aldosterone stimulates the ascending limb of the loop of Henle to make a sodium potassium 2-chloride cotransporter to reabsorb salt from the loop of Henle. Okay, that's AMP2, everybody. Angiotensin, it's aldosterone, has a complicated story upstream that I will not ask you about because that's AMP2. But I could ask you, what cells make aldosterone? The adrenal cortex. I could ask you, what type of receptor does aldosterone interact with? It's intracellular. I could ask you to know that aldosterone is a steroid hormone. That's fair. Okay, so this should be checked for something you should know. This should be checked. Check. Check. And that's it for this slide. But it is important for salt homeostasis by the kidney. So you can see, this is frustrating to me to not be able to get into molecular mechanisms. It's one of the areas where the fact that there's these two courses that are separate is a problem for me, but not for you. I hope those of you who took AMP2 should be integrating. Those of you who didn't should be thinking, oh, maybe I want to take AMP2 after all. I'll learn about salt and water homeostasis. Or not, whatever. Let's talk about calcium homeostasis. You should already know that this is important because of the number of times I've talked about calcium. I mean, how often have we talked about, calcium, neurotransmitter release, hormone release? I mean, calcium, it's important. Contraction of muscle, right? All of these different stories have depended on there being calcium, being enough calcium. How do we control calcium? Two hormones that are in antagonistic control, again. Parathyroid and calcitonin. Parathyroid is a peptide hormone that is released by the parathyroid. The parathyroid are cells that are sort of embedded in the thyroid. So they aren't thyroid cells, but they're with the thyroid gland. We're trying to illustrate them as these colored dots here. So parathyroid means around the thyroid, more or less within the thyroid. They're very different. Parathyroid and thyroid cells are really different, and they respond to different things and they make different things. Parathyroid cells make parathyroid hormone. Thyroid cells make thyroid hormone. The story that I told a few minutes ago. Really different. But anatomically, joined, embedded with each other. Parathyroid cells make parathyroid hormone in response to low calcium in the blood. So if your serum calcium, which is your extracellular calcium, which is the calcium that has to go in through calcium channels in order to do neurotransmitter and hormone release. It's the calcium, it has to get from your blood to your endoplasmic reticulum in order to have calcium release, for muscle contraction. So we maintain blood calcium very tightly. We work very hard to maintain blood calcium because it's so important. The three cell types that are important for controlling blood calcium are kidney, bone, and intestines. We'll see that in a minute. So if the response to low calcium is to release parathyroid hormone, and it causes, so these are the effects, it causes calcium to be reabsorbed from the kidney. We do not want to excrete it in urine. We conserve calcium. We want to absorb all the calcium we can get from our food. So we don't want to excrete it as feces. We want to absorb it as calcium. So it increases the absorption of calcium from food. So we conserve it, we get all we can from it, from our food, and if necessary, we take it from bone. Our bone is a very rich source of calcium. We would prefer not to weaken bone by taking the calcium from it. But because calcium is necessary to keep our amazing nervous systems going, all that neurotransmitter release, if necessary, we will mobilize calcium from bone in order to keep our brain alive. It could lead to osteoporosis. It could lead to weakness and, fractures. This is not what we want, but we will do it if necessary. The opposite of parathyroid hormone is calcitonin. calcitonin is released by parafollicular cells of the thyroid. So again, it's another cell type in the thyroid gland. Not thyroid cells, not parathyroid cells, but parafollicular cells of the thyroid. I'm not gonna nitpick on that one. Different cells in the thyroid. It's also a peptide. It is responding to high serum calcium. If your blood has got too much calcium, what do we want to do? We want to stop absorbing it from food. We want to stop reabsorbing it from bone. We want to, actually, this is phosphate, this is weird, but this is part of excreting it from kidney. Increase excretion, decrease absorption from food, decrease mobilization from bone, and in fact put it into bone, build bone. Sometimes. You may know that vitamin D is an important part of calcium homeostasis. Your parents probably said, drink your milk. It's necessary for growth. Well, it is. It's necessary for growth because it's a necessary coenzyme for bone growth. And I'm not going to test you on vitamin D. But if any endocrinologists heard me talk about calcium without at least mentioning vitamin D, they would string me up by my thumbs. So vitamin D is important for calcium homeostasis, but we're not going to get into the details. I'm not going to test you on vitamin D. It's a hormone. Your body produces it, but you also drink it in vitamin D fortified milk. So you may have heard that you make vitamin D in your skin in response to ultraviolet light from the sun. So you make vitamin D. It's a hormone that you make in your skin. But because some people don't get enough sunlight, it's been put into milk. So if you look at your milk, it'll show that vitamin D is one of the components of fortified milk. Vitamin D. But now, here's another homeostasis problem. Let's see if we can, and this is the last slide, I think. So we have a set point for serum calcium, for blood ionized calcium. Ionized means it's not bound up to some protein. It's free Ca2+, ionized calcium. We have a set point of about 5 milligrams per deciliter, or about, 2 millimolar, something like that. A set point. If the serum calcium goes too low, what hormone is released? What hormone concentration goes up if the serum calcium goes too low? Or similarly, if the serum calcium goes too high, what hormone gets released and what hormone doesn't? So what goes in this box? Well, do this one first. What goes in this box? If serum calcium goes too low, we increase the release of what hormone? Parathyroid hormone. And we decrease the release of calcitonin. Oh. So I screwed it up. Right? Calcitonin goes here, parathyroid hormone goes here. I could give you a problem on an exam asking you to solve one of these homeostasis questions.
Do you want us to also know the set points, or would you be comfortable with just the one?
I would, please do not spend any mental energy memorizing sets. That's not good. You can always look that up.
Okay, thanks.
Okay, yes, we're done for today. Remember, lab this week is to help review for the practical quiz next week. It will be helpful for the leg muscle part of the paper quiz that is due on Sunday, but that's not its primary goal. The primary way you study for the paper quiz, which is this Sunday, is review your endocrine physiology, be able to think about what type of hormone is it? Is it an estrogen? Is it a steroid? Is it a protein? Or is it something else? Where does it come from? What organ produces it? What does it do? What organ responds to it? Those are the ways to study for the quiz, and then study blood muscles. Okay?